There is one in case I'm not loud enough. So what I do today is I'll talk about, I'll eventually get to membrane proteins, I should say. I'll do it on a kind of rather superficial level, just to give you an idea of what one has been able to do and what one can do today in terms of applying bioinformatics to membrane proteins, and what kind of things one can predict about topology and structure of membrane proteins from sequence. So the methodology, the underlying methodology is what you've already heard about. Nowadays, machine learning of various kinds, HMMs, neural networks, basically. So I'm not going to go into the details of that, but I'll just show you how they can be applied in this area. But before going to the real membrane proteins, I will spend some time talking about a very much related subject, which is protein sorting, subcellular protein sorting, and the kind of signals that guide proteins to the correct destination in the cell. And in most cases, these signals guide proteins to be translocated across cellular membranes, either into an organelle, usually into an organelle, or to be secreted from the cell or something like that. And it turns out that membrane proteins use the same targeting pathways and essentially the same translocation machineries that soluble proteins use to be translocated across membranes, targeted to and translocated across membranes. But this also applies to membrane proteins. And so it's an aspect of membrane protein bioinformatics that is important to be familiar with, I think. So I'll spend the first part talking about sorting signal predictions, and then the latter part talking about membrane protein bioinformatics. That's the idea. So protein sorting, if you have basic cell biology, you know, I'm sure you have seen images such as this one, pictures such as this one, that basically make the point that proteins, although almost all proteins are synthesized on cytosolic ribosomes in the cytosol, they all end up in different locations. Some of them, of course, remain in the cytosol, but many go into, many are secreted from the cell, for instance, and in eukaryotic cells, this is a rather convoluted secretory pathway that starts at the endoplasmic reticulum. So the initial targeting event is to target the protein to the endoplasmic reticulum. It gets translocated across the membrane of this organelle into the lumen, and from there it's eventually exported through vesicular transport through the Golgi apparatus to the plasma membrane and eventually secreted from the cell. So that is one targeting pathway, which is specific to extracellular proteins. This is how they're routed. Other proteins are routed for import into the nucleus, for instance. Proteins can also be exported from the nucleus through nuclear pores. Yet other proteins are imported into mitochondria. And mitochondria have two membranes, so some of these proteins have to be imported across two lipid bilayers actually before they reach the final destination. There are other organelles such as peroxisome that have specific targeting signals and machineries that recognize the targeting signals and translocate the proteins into the peroxisome. And of course in plant cells you have chloroplasts. And most proteins in the chloroplast are actually synthesized in the cytosol and then imported into the chloroplast. The chloroplast, in fact, has three membranes, so some chloroplast proteins have to be translocated across three membranes before they reach the final location that they're supposed to be at. And all of these sorting events are essentially depend on sorting signals present within the protein itself. Of course, they look different for the different compartments, but the basic principle is that the sorting information is encoded within the polypeptide itself in various ways. So in the cell, there are various kind of signal receptors that can recognize these particular signals and target proteins to the correct destination. Sometimes post-translationally, that is to say after the protein has been fully manufactured, and only then is it targeted and imported. But in other cases, such as the endoplasmic reticulum, this is a co-translational process. So the targeting signal in that case is at the very end terminus of the protein. It emerges from the ribosome, the very first thing that happens. And then the whole ribosome complex is targeted to the membrane of the endoplasmic reticulum. So it can then secrete the protein continuously or translocate the protein continuously across the membrane as it's being manufactured. That's called co-translational translocation. So this differs between different targeting pathways. But in every case, there are then proteins that can recognize these targeting signals wherever they're located in the chain, protein chain. So the cell can recognize these signals, has machineries to recognize the signals. And then you would think, well, then as bioinformaticians, you should also be able to recognize these signals. You should be able to extract the relevant features from the sequences, and then use those in prediction programs to basically do what the cell does, to figure out from any given polypeptide sequence where it's going to end up in the cell. So that is the bioinformatics problem. Given a protein sequence, predict where it's going to end up in the cell, which organelle. So I'll mainly talk about one of these sorting pathways, and that is the secretory pathway, where the initial targeting event is that an N-terminal part of the protein is recognized by receptors targeting the ribosome that is manufacturing this protein to a translocation site on the ER membrane, or a translocation channel in the ER membrane, whereupon the protein is then translocated into the lumen of the ER as it's being synthesized. And in fact, this signal sequence is then clipped off at a certain stage when it's no longer needed, when the targeting has happened, then it's no longer needed and can be clipped off. And eventually the protein is then routed through vesicular transport to the plasma membrane, and the protein is secreted. So that's how it works. So then, what do signal peptides look like? The signal peptides that target proteins to the endoplasmic reticulum. Now this is a very old bioinformatics problem. And it was one of the first, really, in sort of molecular biology, one of the early examples of bioinformatics predictors being developed. And of course it depended on the ability to sequence secretory proteins to find out what the signal peptide looked like. And then hopefully by comparing many, finding some common features that would allow you to predict that this is a signal peptide, this is not a signal peptide. And this goes way back to the very early days of DNA sequencing, where one did what's called cDNA sequencing, basically extracting mRNAs from cells, using a reverse transcriptase to turn them into a DNA sequence, and then actually people cloned these DNA sequences into plasmids, and then finally sequenced them, so that the entire protein sequence was available, including this N-terminal bit that has the signal sequence. Now one couldn't do this from protein sequencing, because once the protein was exported, the signal peptide had been removed already. So there was no good purifying the protein from blood, for instance, and trying to sequence it on the protein level. One had to go to the DNA level to do this. One reason why this was kind of an early success, if you will, was that this signal sequence, this targeting signal, turned out to be quite distinct and quite easily recognized by just looking at the sequence. So already I think the first papers were done with maybe 20 sequences or so of signal peptides. So the very first cloning papers, basically, where one could read off signal peptides. And it turned out that even with just 20 or 30 sequences was enough to do a sort of a reasonably good predictor for signal peptides. And now, of course, as I will show you, there are many more sequences available, and so you can train predictors in a much more efficient way, and obviously get better performance. But what was clear already from just a few sequences was that signal sequences, either from bacteria, which can of course also secrete proteins and use a homologous machinery to do it, compared to the eukaryotic machinery. So both in bacteria and in eukaryotes, they pretty much look the same. They were not well concerned on the sequence level. It wasn't just a single particular sequence of amino acids that encoded a single peptide. Rather, they're very variable in terms of sequence, but they all turn out to have a distinct kind of three-partite structure or design that was common to all of them, even if the precise sequence was different within every one. And this was quite quickly realized that essentially all of them start out with a few positively charged residues right near the N-terminus, then a hydrophobic segment, typically sort of 10, 12 residues long, and finally a short region of 5 to 6 residues that leads up to the cleavage site, where a specific enzyme called signal peptidase will actually cleave it off at some point. And what was immediately realized was that the amino acid right before the cleavage site and 2 positions upstream of that seemed to require, one always found that essentially small residues in these 2, like lysines and alanines, but never big bulky residues. So apparently the enzyme would recognize this kind of peptide with 2 small amino acids and be able to cleave it in a specific site. So this was apparent, this kind of general design was apparent already with just a few sequences, and could be turned into an early predictor, and you can see how old this is. 1986 was when this was actually first published. The very first kind of reasonably well-performing method, and it was a very trivially simple algorithm to do it. It was based on what's called the weight matrix, which is essentially, so what was done here was to take, well actually this was based on somewhat bigger data set, maybe about 80 or maybe about 100 sequences or so. So basically they were all aligned from their cleavage sites, so the cleavage sites were all aligned, and then it was just counted how many amino acids, how many alanines, for instance, were found in each position relative to the cleavage site, right? And how many cysteines and aspartates and so on. And so here is the cleavage site between -1 and +1, and you can see alanine is very abundant in -1, 80 examples. Glycine is highly abundant, 39 examples, and then serine is also quite abundant, right? But you see in this column here, you see a lot of zeros. So there are many residues that were never found in that -1 position. In -2, it's more even if it's still not completely random, but it's a more even distribution. But then again, in -3, you see a lot of zeros, and it's again mostly small residues that are seen in -3 position. Right? So that's what I told you. If you go further in, you start to see more hydrophobics like leucine enriched in this region relative to the cleavage site. And as you go really far to the N-terminus, which is probably too far to be included here because it only goes to -13, you would start to see positively charged ones like arginines and lysines, but that would be out here somewhere. So these counts is just giving you the counts, and this can be turned into a weight matrix by just taking the log, essentially. So you take the number of, say, alanines observed in position -1, and you divide it by the average, or the average and you divide it by the average number of alanines across the whole, matrix, to give you an idea of whether it's enriched or not enriched in a particular position. So you take the quotient and you just take the log of that, and you convert this then into a weight matrix. And then you can make a very simple prediction method by just scanning along the sequence with this weight matrix, and for each position of the weight matrix in the sequence, you just sum up all these weight values. And that will give you a score for that particular segment of your sequence, how much it looks like a signal peptide, right? Then you move it over by one residue, and you repeat the calculation, and you move it over and move it over. So for each sequence, you basically get a set of scores, and if there is a signal peptide in that sequence, once you put the weight matrix exactly to coincide with that sequence, so the -1 and the +1 positions coincide, then you get a high score, because you'll get very good fit to all of these weights, right? And then as you move further on, it's going to go less again. Right?
So, is the cleavage site very precise? Or is
yes.
Okay. I mean,
it's very precise. Yeah. I mean, there are few examples with heterogeneous N-termini where the cleavage site could be, differ by 1 or 2 amino acids, but in most cases, it's very precise. Yeah. So that was, in those days, that was the most advanced bioinformatics you could do. And here is just a compilation of scores where of the maximum score as for each sequence as you run the weight matrix along the sequence for a bunch of just cytoplasmic proteins that don't have signal peptides. Yeah?
We were wondering what the expected value is for. Is it like an average over the whole signal peptide?
Yes.
Okay.
Yeah, over the whole matrix actually. What's the average frequency of alanine across all these positions, right? So yeah. So this distribution here is for, say, 100 cytoplasmic proteins that don't have signal peptides, and you take the maximum score from each one. And this is from a similar size sample with signal sequences in it. Right? So it was, for this very limited data set, this was a fairly clean separation between, you could achieve a fairly clean separation between the two. But of course, this was just reapplying the algorithm to the original data set. So it wasn't jackknifing or anything like that. It was just so clearly, when new sequences appeared that were not in the original data set, the performance would not be as good as this, as with all prediction methods, essentially. But still, it was a start. It didn't have very much practical use because there were so few sequences, and it helped to some degree to predict cleavage sites, basically. But it was interesting from kind of a bioinformatics perspective, anyway. Then what happened was that machine learning came into the picture, and the most modern, all methods now in this field use machine learning. So this was a review of that, but basically, what turned out to be very useful in this particular problem was neural networks. And I'm not going to go in. I mean, you've already seen neural networks there. Right? So I think you can easily imagine how you can take, if you have a sample of signal peptides with annotated cleavage sites and another control sample that are negative examples, how you could use that to train a neural network, right? So this was done initially with a couple of theoretical physicists in Denmark that had realized that would be interesting to apply machine learning or neural networks in that case to sort of biological sequence problems. It's one of the early uses for neural networks, although I think they came from speech recognition or something initially. But they were applied early on to secondary structure prediction in molecular biology. I think that was the first application neural networks, and this was a rather early application as well. So input layer, output layers, hidden layers. You train it on a high quality data set, of course, positive and negative examples. You can try different architectures, and then you just optimize the parameters and evaluate using cross-validation and jackknifing. In the early days, of course, the positive and negative examples were picked by hand from the literature. And so the data set was actually very high quality because every paper had been read, every paper where the protein or the gene had been sequenced was read before it went into the training set. One really checked, is it really known experimentally where the cleavage site is, yes, no, etc. So although the training sets were small, they were very high quality, actually higher quality than what they are now when we just take them from databases. But of course, it was small data sets. So this led to what was initially called the signal P predictor, and this has now been around for a long time, and it's become a whole family of signal P predictors that have been updated regularly and retrained or trained using new architectures and so on. But this was the original, or one of the first signal P, well, this was actually signal P 4.0 already, but the previous ones were very similar. So looking at an output from a signal P predictor is kind of instructive, I think. So this basically, at least in the early versions of signal P, 3 different scores were computed from 2 different networks actually. And one network was trained to predict cleavage sites, not care so much about what was outside the cleavage sites, but to focus on trying to predict the precise cleavage site. That's called the C-score. And when that network is applied across the sequence, which actually does have a signal peptide in it, you can see a few, well, at least one positively charged residue here, arginine on the N-terminus, and then a hydrophobic stretch, and then there is a cleavage site hidden in here. In this particular case, the predictor identifies this as the cleavage site between the, I guess it's between the, yeah, I think it's between, it's VTT, I think it's between the T and the E that's predicted as, it should be here somewhere. Yeah, position 30. Yeah, it's just between the T and the E here. That should be that's the predicted cleavage site, and you see that has a higher score than anything else around it. Then there is a second network, the green one, called the S-score, and that's just, it doesn't look really specifically for the cleavage site, but it looks for these other features of the single peptide, the positive charge N-terminus and the hydrophobic region. And so it basically scores, it gives a high score throughout this initial N-terminal region, basically, and then it drops down to kind of background levels, basically saying that, yeah, it looks like you have a single peptide here, and the cleavage site predictor then says, and it looks like it will be cleaved at this particular site. There's also a third score, that the Y-score, that was used initially to improve the cleavage site prediction. And the idea there was that although this green predictor is only kind of generally scoring the signal peptide likeness of the sequence, you see that it drops down at some point. And the idea was that if you have multiple possible predicted sites for cleavage, here is only one, but you can imagine it could be a few more, right? Then the most likely one is probably where this green network drops from a high value to a low value. That should roughly be where the most likely cleavage site is. So what the Y-score is essentially a derivative of the green score. So it identifies the region where you have the largest drop in the green score, and then it focuses, it essentially weighs the red scores with this derivative, so that you kind of focus on this region where to be the most likely region to look for a cleavage site, and that improved the cleavage site prediction somewhat. So So that's how these signal P predictors, the early ones at least, were set up to work. And they actually performed already at this stage, performed really well. And so they were, and still are, I think, incorporated into all the annotation pipelines and Uniprot, I think, still uses one or other version of signal P to predict key signal peptides in DNA sequence data, in genomic data, and so on. So it's been very widely used. And it's, like I said, it's been updated regularly with more sequence data, retraining, improving the architecture, etc. So I'll just show you the latest version that came out last year, Signal P 5.0. And now it's becoming rather complex, right? Now the guys doing it were, deep learning had to be applied to this problem, obviously. So they came up with this deep recurrent neural network, and you see it has all kinds of, I won't even try to pretend that I can explain all this to you. But it's all kinds of forward and backwards networks, and also the data sets have been expanded so that there are specific data sets for different kingdoms. So there's a bacterial data set, there's an archaeal data set, there is, I think, a yeast and a eukaryotic data set. So they've been trained on separate data sets. And so you also input from where the sequence, what kingdom the sequence is from, and so on. And also the training set is, if the first one was about 100, this is now 3,500 signal peptides that go into training of this. In fact, in signal P, there is also a couple of other types of signal peptides included. So it can predict, I think, 3 different types of signal peptides that I won't talk about how they come into the picture, but it can start to distinguish between different types of signal peptides as well. But the basic user interface looks almost identical to all the early versions. You basically just paste in your sequence and run it, or you paste in a set of sequences and run it. And just to give you some idea of performance here is, so these are three different types of scorings, in blue, orange, and green. This is Matthew's correlation coefficient that kind of weighs together false positives, false negatives, and true positives and true negatives in a combined score. So it kind of gives you the overall, tries to find a compromise score that balances false positives, false negatives. And then it's recall and precision scores for cleavage site prediction. So the precision score is the fraction of cleavage site predictions that are correct. So you predict 100 cleavage sites. How many are correct? That's what fraction is correct. That's the CS precision. And the recall is a fraction of real signal peptides that are predicted as signal peptides and with the correct cleavage site. And you see signal P5, signal P4, and another predictor called DeepSig. And you see there are slight improvements in signal P5 compared to the older ones. In terms of Matthew correlation, it's not huge. In bacteria, it's almost the same, slightly better in eukaryotes. But it's doing a bit better, especially on the precision measure, but also on the recall. So with every iteration of the signal P predictors, they become a little bit better and been able to, in this case, predict a few more types of signal peptides as well. But I think this, from a sort of bioinformatics perspective, I think you can say this is a mature field. It's just for the people who have spent their whole careers working on this. It's worthwhile to maybe improve it again, but it's not something that a young bioinformatician will go into, I think, because it's there is a little room for improvement, but it's not clear that just training on more sequences is going to fill in the final percentage points here. But overall, you get correctly predicted signal peptides with correctly predicted cleavage sites in about, let's say, 80% or so of the cases for bacterial signal peptides. Okay, so this is signal peptides for protein secretion. But as we saw on that initial slide, there are signal peptides, other types of signal peptides that target proteins for other organelles simply. And at the end of the day, what you would like to have, of course, is one master predictor that can do all these types of different signal peptides and essentially distribute all the proteins to all the correct locations in the cell. Right? So, and there are a few attempts to do that. One is, again, from the same people in Denmark and here in Stockholm, called Target P. So Target P is basically collection networks, each one predicting one type of targeting event, but then weighed together so that, basically what Target P does is it says, well, given all the predictions, individual predictions for mitochondria, for ER, for chloroplast, actually, yeah, chloroplast is in there as well, which is the most likely one? So it's trying to answer that question, right? And it distinguishes between 5 or 6 different locations. Then there is another one developed in Japan that has many, many more possible locations. So for instance, Target P would only say, well, this protein has an ER targeting sequence, an ER targeting signal peptide. But this more advanced one would actually say, well, it has a signal sequence on it, so therefore it's going to be targeted to the ER. But then it looks in the rest of the sequence to try to predict whether it's a protein that's going to stay in the ER, a protein that's going to stay in the Golgi, for instance, or go to the lysosome, which I didn't show here, or eventually be secreted. So there are kind of sub-locations within the secretory pathway where proteins can be retained. And they also try to predict that, right? Or the same thing with mitochondria. Is the protein predicted to end up to go through 2 membranes or only through 1 membrane? So there are sub-compartments within the mitochondria that are addressed in this other more complex predictor. Of course, when you have many more locations, you have many more possibilities to make errors, and so the overall performance goes down. But it's still possible to try to do almost what the cell does in this field and move, and basically you can essentially take all the proteins predicted from a genome sequence and run it through here in one of these predictors and get some reasonably good idea of where all the different proteins are going to end up in the cell. But not with the precision the cell can do it. So this was signal peptide prediction, or targeting peptide prediction. And it's been a subfield, as you saw, from the mid-80s in bioinformatics, and it's still a little bit of activity in it, but the methods have more or less reached probably the limit of how good they can become. And the connection between this and membrane protein bioinformatics is that, again, if you go back to the cell, so membrane proteins, let's say plasma membrane proteins, they are also targeted through the secretory pathway. So they are initially targeted to and actually inserted into the membrane of the endoplasmic reticulum, if they're destined for the plasma membrane, for instance. And they use essentially the same targeting machinery and the same translocation machinery that the secretory proteins use. So they also have to have a signal sequence, but in opposition to the secretory proteins, they're not completely translocated across the membrane, but parts of the membrane protein stick in the membrane, right, and become transmembrane. So from a biosynthetic point of view, it's membrane protein biosynthesis is very much connected to this general problem of protein targeting and protein sorting. Same thing for mitochondrial membrane proteins. Actually, many of them are encoded within the mitochondrial genome itself, but not all. And those that are not encoded within the mitochondrial genome, they have to be targeted through the normal protein import machinery in the mitochondria, and only then get inserted into the membranes of the mitochondria. And the same with these other ones. Actually, membrane proteins from the nuclear membrane also go into the ER initially, because the ER, it's not shown here, but the ER and the nuclear membrane is a contiguous membrane system. So you can actually diffuse in a bilayer from the ER into the membranes of the two membranes that enclose the nucleus. So nuclear membrane proteins also go to the ER initially, and they're inserted there, and then they're directed to the nuclear membrane. So therefore, it was kind of a natural thing that people who've been working on signal sequence prediction, like myself, got interested also in membrane protein prediction, because they're biosynthetically, they're very similar. They use the same machineries and so on. So I guess we'll take a break in a few minutes, but I'll say I'll just introduce membrane proteins a little bit, so then we can focus on the more bioinformatics aspect of it. So of course, membrane proteins from a sort of biophysical perspective or physical chemistry perspective obey completely different design principles than soluble proteins. And that is because they are designed to sit happily within a lipid bilayer rather than diffusing around in an aqueous solution. So on this MD simulation, actually, you see the lipid bilayer with the lipid head groups facing the surrounding water, and the lipid tails, the hydrocarbon tails, hydrophobic hydrocarbon tails in the lipids, basically forming the core of the lipid bilayer, right? Expelling all water molecules from these hydrophobic lipid tails. So of course, lipid bilayer is formed spontaneously in water. If you have the right types of lipids and the right pH and so on, they will assemble spontaneously, because that's how you essentially minimize the exposure of these hydrophobic carbon tails to the water molecules. You basically shield the hydrocarbon tails by having these lipid head groups provide the shield on each side to interact with the water molecules. Now, generally speaking, these lipid bilayers are really barriers to the diffusion of polar molecules. So if you had a cell that was surrounded by a pure lipid bilayer, it would not be able to take up ions or expel ions or whatever polar molecule or charged molecule you have, would not be able to penetrate into the cell or be expelled from the cell. Water molecules, to some extent, would be able to penetrate. They have very low penetrance, but on the other hand, they're at very high concentration, right? So it's in pure water, you have, I think the concentration is 55 molar or something of water. So even though the penetration through lipid bilayer is is very small, and you see in this simulation, of course, the water molecules don't make it into the lipid bilayer. But if you have a very large bilayer surface and a very high concentration of water molecules, eventually some of them are going to get across. In this particular simulation, the protein, this blue thing, that has been put into the lipid bilayer is actually a water channel protein. And that's why you see these water channels, or these row water molecules that actually do penetrate the membrane with a fairly high frequency. But that's because this protein is specifically designed to provide a water channel across the membrane. And of course, other membrane proteins are there to allow ions in and out, or to pump ions or small molecules from one side to the other, etc. So what's typical about membrane proteins then is that they are, or at least big portions of them, are designed to be very comfortable within a lipid bilayer, but not when exposed to water. So they will essentially be driven into the membrane, just as lipids are driven into the membrane. Right? Now that we have quite a large number of structures in membrane proteins, although they lag behind soluble proteins by a factor of probably 1,000 or so, if you just count the numbers of known membrane protein structures and known structures of soluble proteins. But we still have quite a good inventory of high resolution structures in membrane proteins. And what one finds then is that they essentially come in only two basic architectures, two basic designs for integral membrane proteins. And one is like the one that was on the previous MD movie, is where the transmembrane segments are folded into alpha helical rods, and of course are composed mostly of hydrophobic amino acids, right? So if you have a stretch of hydrophobic amino acids, you coil it up into an alpha helix, that helix is going to be a nice hydrophobic surface, right? With the side chains, the hydrophobic side chains dressing up the helix, basically. So it's easy to imagine that a stretch of hydrophobic residues coiled up into an alpha helix would be very happy inside the lipid bilayer. The other design is what's called the beta barrel design, and here the chain is in an elongated conformation, in a beta strand conformation, and the whole thing is coiled up into a cylinder or a barrel. So there is one basic principle that can explain both of these types of structures, and that is to take care of all the backbone hydrogen bonds that can form within a polypeptide. Right? So if you just have an extended piece of polypeptide, even if the side chains are all hydrophobic, it would still not be very comfortable spanning a lipid bilayer, because the backbone hydrogen bonds would not have a partner, right? From the carboxyl groups and the NH groups. If you keep it in water, the hydrogen bonding is fine because it can make hydrogen bonds to water molecules, but of course the hydrophobic residues would be unhappy. If you stick it across the membrane and maintain this extended conformation, it would be the opposite. The side chains would be happy, but the hydrogen bonds would not be, because you would have to strip off all the water molecules, and so you'd have these unsatisfied hydrogen bonds within the membrane itself. Now, one way to solve that problem is you take your strand, extended strand, you coil it into an alpha helix, because in the alpha helix, all the backbone hydrogen bonds are satisfied internally, right? From one turn of the helix to the next turn of the helix. So all the OHs, all the carboxyls and all the NHs are now engaged in hydrogen bonding within the alpha helix itself. And so now you have a nice hydrophobic rod-like thing, no hydrogen bonds, no unsatisfied hydrogen bonds, just a hydrophobic surface that would fit very nicely across the lipid bilayer. And in the beta barrel architecture, the problem is solved in a different way. So here, if you again imagine one of these strands, of course, if you put it into the membrane again, you'll have all these unsatisfied hydrogen bonds. But if you imagine a large planar beta sheet, so you have all the strands, let's say it's an antiparallel beta sheet, so you have antiparallel pairing between the strands. As long as you focus between strands, all the hydrogen bonds are again satisfied, right? All the backbone hydrogen bonds are satisfied within the beta sheet itself. Except if you now put the beta sheet into a membrane, it will be fine from the point of view of hydrogen bonding, except at the edges, right? So you'd have one strand at this age, one strand at that age, that would have hydrogen bonds that are not satisfied. So the simple solution then is to basically fold that into a barrel and let the first strand and the last strand pair up and make their hydrogen bonds. So now you have a barrel, all the hydrogen bonds are internally satisfied, and all you need to ensure is that you fold up the beta sheet in such a way that you create a hydrophobic surface on the outside, meaning every second residue in each strand should be hydrophobic and point towards the outside. So then you would form a nice barrel-like structure, again with a nice hydrophobic surface and no hydrogen bonding problems that you could now integrate into the membrane, right? And that's exactly what happens in the beta barrel product. On the inside of the beta barrel, the residues that project into the barrel, they could be anything. They could be hydrophobic, they could be polar, they wouldn't be exposed to the lipid regardless. So they can be anything. So you can manipulate the inside to make pores or channels or what have you inside these beta barrels. And of course you can manipulate the number of strands that go into the barrel. This is a very tight barrel, I think with 8 strands or so. That's about as tight as you can make it. If you try to make it with 6 or 7, you wouldn't actually be able to fold it up because it would be too crowded in the middle. But 8 is doable, and there are beta barrels that have, I think, 24 or even more strands in them, and then of course the center is quite wide. So you can even imagine taking quite big molecules across these really big channels. But these are the only 2 ways actually that nature seems to have been able to solve the problem of making a continuous hydrophobic surface without having the problem of exposed hydrogen bonds. So it's either helix bundles or beta barrels. And essentially every membrane protein can be classified into one or the other type of architecture. But of course, beyond the architecture, they can have very different folds and perform very different functions. So I think that's where it's time to take the break, and then we will go in to look at what bioinformatics has been done, especially on the helix bundle proteins, but a little bit also on the beta barrel.
Do you mind if we make it a short break?
Yeah, sure. We can
like
just
5
take
minutes?
5 minutes if you want.
Thanks.
I just want to
Okay.
make a quick trip,
Yeah.
and then we can leave. We can stop early for sure.